Hello, everyone. Today I will be presenting about the work that I've been doing on the profiling of the T-cell repertoire in patients with non-small cell lung cancer to evaluate their predictive value for response to immunotherapy. And this study has been supported by the OncoMind Clinical Research Grant. And here are my disclaimers, in particular that this study has been funded by Thermo Fisher Scientific, and I also have received an honorarium in relation to this presentation. In general, is now a very well-known established treatment for non-small cell lung cancer, the use of anti-PD-1 therapy. And this, in the case of the single-agent treatment, has been based on the expression of PD-L1 in the tumor cells. However, it is known that the combination of pembrolizumab with chemotherapy, based on the results from the Keynote 024, 42, and the Checkmate 227, that there is the combination of immunotherapy together with chemotherapy results in better outcomes and survivals independent of the expression of PD-L1. However, patients with high PD-L1 still perform better than patients with low PD-L1. And this remains the main biomarker study for clinical decisions in the context of PD-L1. But there is an overall interest on studying other biomarkers that can indicate the benefit for combination treatments and also for identifying patients that will require additional therapeutic investigations due to the lack of probability of treatment of success from immunotherapy. Amongst the biomarkers investigated are the tumor mutational burden have shown a high to be indicative also of better outcomes, microsatellite instability, and the T-cell infiltration, the presence of tertiary lymphoid structures, as well as the presence of other immune checkpoints. But in particular, our team have been interested to understand better the central interaction that drive the immune response. And this is the presentation by the HLA molecule of the antigen, the new antigens presented in response in the context of a tumor, especially new antigens derived from the tumor mutational burden, and on the other hand, the existing T-cell response that have already been mounted against the tumor in patients that will be recognizing these antigens. Therefore, we've been investigating these three aspects. Some of our studies have been focused on the HLA analysis, and we have shown in previous publications that the presence of maximal heterozygosity, so it's patients that don't have any homozygosity in any of their HLA loci, have a better outcomes from a singular agent anti-PD-L1, and certain supertypes also are associated with better with the survival of non-small cell lung cancer patients treated with immunotherapy. On the other hand, HLA heterozygosity is also associated with the development of immune-related adverse events, and so that's been the subject of previous studies. We also have ongoing studies on the analysis of the tumor mutational burden on these patients and their association with response to treatment. And pertinent to this presentation is the analysis of the T-cell response. And we in particular want to know what the T-cell repertoire of these patients prior commencing therapy, and the analysis in circulation as a more accessible sample from which to analyze the repertoire compared to actually uptake a sample from the tumor, which might not be always possible in the case of patients treated with immunotherapy, with patients with non-small cell lung cancer. Okay, for this particular study, we selected 128 patients with advanced non-small cell lung cancer, 63 treated with combination singular agent pembrolizumab and 65 with pembrolizumab plus chemotherapy. From them, we retrospectively narrowed down to 48 patients treated with pembrolizumab and 53 with pembrolizumab chemotherapy after exclusion due to different clinical and analytical factors. For this analysis, we carry on with particular focus on the analysis of the TCR beta, and we use the SR assays. The focus on TCR beta is justified by the fact that this is the most diverse chain within the TCR alpha beta receptor, which is usually the one involved in the recognition of antigens by the CD8 cells, and in particular because we are interested in short fragments for twofold. One is the number of samples that we can put in an on-command chip, so we can analyze a larger volume of samples. And second, because it's less dependent on the quality of the DNA, which enables us to then do comparisons between the repertoire in blood and the repertoire in tumors from tumor-derived genomic DNA from FFP blocks. For the analysis, we extract genomic DNA from white blood cells, which then we quantify, and we input into our libraries 200 nanograms of genomic DNA. For library preparation, these libraries then were sequenced in Ion Studio S5, and the initial analysis was done in an Ion Reporter software. For analysis of downsampling of the whole data set, there was a plateau of the number of clones observed from 240,000 cutoff for downsampling. However, we analyzed our data 1 million downsampling, which conform for most of for all our samples to be included on the analysis. And we focus on the analysis of 4 different metrics of the TCR repertoire. And these are, I hope, is better represented in these diagrams. One is richness, which is the number of unique clones. The other one is evenness, how well these clones are distributed, and you can get a large number of clones, but they might not be evenly distributed, and vice versa. And Shannon diversity is kind of a combination between these two. It combines the metric of richness as evenness and provide a better representation for the diversity of within a population of sequences. Convergence, on the other hand, is a different type of metrics, and is basically the representation of how many of clones with different nucleotide sequence actually end up with the same amino acid sequence. And this is a very insightful measurement because it does provide an idea of that there is a pressure in selecting certain protein sequences, which ultimately are the functional part of the T-cell receptor. So we carry on the comparison of each one of these metrics across responder and non-responders in both cohorts. When we did this comparison, the main difference observed was the difference in between the number of clones on patients treated with single-agent pembrolizumab, with patients with non-responder have higher number of unique clones than responders. None of the other metrics were significant show significant difference between responder and non-responders across both cohorts. Then we look at survival, whether the TCR repertoire had any indication or association with the outcomes, survival outcomes in this patient, in particular progression-free survival. And we observed that in a cohort treated with pembrolizumab and plus chemotherapy, there were significant differences in the survival of patients depending on the TCR metrics, with patients with higher number of unique clones, a higher Shannon diversity, having longer progression-free survival, while patients with a lower evenness and lower convergence had also significant longer progression-free survival. We carry on this analysis also for overall survival and observe that there is a significant increase in longer survival in patients treated with pembrolizumab plus chemotherapy in relation to the number of unique clones. And this was in particular the most distinguished metrics. Then we decide to, instead of using this metric as a separate factors, to combine them into a logistic regression model. By combining them into a logistic regression model, we observe that an improvement on the area under the curve on the distinguishing respondent from non-responders by combining these metrics, and the model score basically provide an area under the curve of 0.693 for patients treated with pembrolizumab and 0.760 for patients treated with pembrolizumab plus chemotherapy, with a clear difference in the model score between responder and non-responders for both cohorts. And this classification also was validated using an intrinsic cross-validation by splitting the training and testing sets, multiple with bootstrapping, and showing the consistency between the area under the curve on the graph to the right. We then took this model score and classified evaluate patients with high and low model scores in relation to their progression-free survival and overall survival. And what was interesting here, then now we observe a clear segregation between patients with high model score have a benefit in progression-free survival in the case of the pembrolizumab cohort, and a statistically significant increase in the length of overall survival for patients treated with pembrolizumab plus chemotherapy, represented by the Kaplan-Meier curves observed on the right. We then combine the model score category classification together with the predictive value of other well-known metrics that have been associated with response, such as the ECOG of score of these patients, the age, and neutrophil to lymphocyte ratio. And we observed that in this multivariate analysis, indeed, the model score was shown to be an independent predictor of response, of survival in both cohorts, in patients treated with pembrolizumab and patients treated with pembrolizumab plus chemotherapy. In addition to that, we evaluate these metrics in the context of the prediction of potential toxicities or immune-related adverse events. However, we did not observe any differences between patients that developed immune-related adverse events and those that did not, in association with the profile of the CDR3 repertoire. So in conclusion, we observed that the number of unique clones may be associated with improved clinical benefit in patients treated with single-agent anti-PD-1. And in the case of patients treated with combination immunotherapy, the TCR repertoire metrics may be more informative, and they may be associated with longer progression-free survival in particular. And all of them, either increasing unique clones and increasing diversity, was associated with longer progression-free survival, as well as low evenness and low convergence. However, only unique clones were found to be associated with overall survival in the context of patients treated with anti-PD-1 and plus chemotherapy. The more informative part of this is combining these metrics into a model score, which was has shown a very strong classification of responder and non-responders in this cohort, and in particular the prediction of overall survival in patients treated with anti-PD-1 plus chemotherapy. These positive results kind of are conducive to support the validation of this finding in prospective studies, we can indeed demonstrate that that the TCR profile matrix can be used to identify patients that are more likely to benefit from immunotherapy or combination immunotherapy in a context of non-small cell lung cancer. Another of our follow-up studies will be done, analyze the same type of metrics, but in the context of a metastatic melanoma. With that, I would like to thank the people that are carry out this work, in particular, Dr. Afaf Abed, which is an oncologist and PhD student with my team, Aaron Beasley, which is the one responsible to do the NGS sequencing as well as the bioinformatic analysis, and the rest of my team and the clinical team that work with me, the study participants, and of course our funders. Thank you.
And once again, we are back live here for our Q&A session. I would like to thank again Dr. Gray for her excellent presentation. So Dr. Gray, it's great to have you with us. Here comes the first question for you. Thanks for joining. So, I mean, your results are honestly striking. Some of the correlation that you have found up, seems to be extremely solid. And the question now goes into the direction, are those, in your opinion, purely related to the agent that it was used? Or do you think those results could be derived as well for other checkpoint inhibitors?
Yes, that's an excellent question. I believe it will be relevant to other checkpoint inhibitors because it's really defining about the existing T-cell repertoire that the checkpoint inhibitor will unleash. So it is possible that, for example, with chemotherapy, we thought that the chemotherapy will create de novo antigen recognition, and therefore there will be less apparent that the pre-existing repertoire already will signify the efficiency of the response to combination pembrolizumab with chemotherapy. But however, even in that setting, actually performed very well. So I think it tells you something about that the pre-existing repertoire of T cells, it carries some information about the capacity of the immune system to see the tumor. Whether it's specific to the tumor, and we're doing some other studies that are more clonal in nature. It's a different question, whether it's something more about the immunostatus rather than a specific anti-tumoral T-cell response. It could be queried, but I do think it will be relevant for other immune checkpoint inhibitors, which that was the question.
Yes, and thank you very much for answering that. I think that's really a very important one, as it will open up for the opportunity of having, different kind of treatment to be analyzed with the same biomarker being relevant for all of them. So again, thanks for answering. Here comes your second question. And it's actually about possible future implementation, okay? So how complex is, in your point of view, in your experience, the actual utilization of this TCR solution? So do you think it is feasible in a not too far away future to move it farther down the line and getting it closer to the routine testing, or do you think it's still a too complicated test to be implemented in routine?
Yes, so one of the discussions when we designed the study was whether we just go with a simple. This is a relatively simple method. You can just use the Buffy code of collecting from an EDTA tube, extract DNA through a system that could be in a robotic system, and then put it through the Thermo Fisher pipeline. The new systems can set up the Genoplex, can set up the whole, even library preparation doesn't have to necessarily done it manually, and it's a really straightforward process. And the reason why I bring that up is because in parallel to that, we are also more interested in clonality. So a lot of our project proposals and parallel projects are to go into the PBMCs, look for single cell sequencing, look for which TCR clones actually recognize HLA presented antigens, which is beautiful for science from the scientific perspective and the inquiry, but possibly not that relevant for a clinical application in the short term because it's a much more complicated process, a much more costly. This approach, the clinician that was leading the study, Dr. Afaf, was very keen on doing it in a relatively simple pipeline, because she says if this works, then it's much easier to implement and to have it run routinely than a single cell analysis.
All right. Well, thank you very much again. I think this is, in a way to say, still more work to be done. There's still some times to go before a full uptake into the routine testing, but yes. Very solid data. Yeah. Thank you.
Sorry, yes,
very solid data.
The data need to be validated. So there is two aspects to it. I think the pipeline is very simple and easier to implement. I think what is important is to, on top of that, then have a good validation, a multi-center, cross-validation, independent, and this then will set up for going into routine test, more applicable to routine testing. But the analysis pipeline is actually simple, and that's what we like.
Thank you very much for that again. So I would say then we look forward for this additional multi-center study. I'm very convinced TCR is definitely a key to make sure that immunotherapy full value is unleashed for the patient. So we look forward again to move what it's now a translational research tool into something more usable for routine testing, but that will require some additional work. Again, thank you for your presentation. So now we will take a 15 minutes break, but please take the chance to explore the Oncomind World environment and be back with us at about, again, 15 minutes, so it will be 35 from now. And again, thank you, Dr. Gray, for participating in this event, and I look forward to the next session. So talk to you soon.
Thank you. Thank you.